Tēnā koutou katoa. Kia ora e te whānau me ngā rangatira e noho tā wāhi ana. No Ingarangi, no Kotirana o Kutupuna, ko Oxford te whenua tupu. Ko Salian Harbison aho. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you to the Royal Society Te Aparana for this great honour, to ESR and the University of Auckland, and especially my forensic colleagues and friends far and wide. My personal journey includes crime scene to courtroom and the innovation of technology to underpin forensic science services. And today we're going to talk about some applications of forensic genetics to crime investigation. First of all, how do we use biology in forensic science? First of all, we identify and collect samples from a variety of cases and from a wide range of things. We identify the biological source of the material using chemical tests, RNA, and then we make DNA profiles. We compare these DNA profiles from crime scenes and objects with those from people, or we compare DNA profiles from unknown people with the DNA profile databank. Then we carry out some statistical interpretation and present our findings as expert witnesses in court. And as technology advances, so does forensic research and its applications. First of all, let's talk about the source of the material, body fluids, and its relationship to DNA. So DNA codes for genes, and genes are transcribed into what is called messenger RNA, which is the instructions the cells need to build the proteins that make each cell specific and special. So mRNA is the key to identifying different types of cells. We discovered in a light bulb moment that mRNA was stable in dried body fluid stains. And we used this discovery to design mRNA profiles to detect the unique messenger RNAs present in blood, menstrual blood, semen, saliva, and vaginal material. We also integrated this with the DNA profiling process in use in the laboratory in order to save the precious evidential material. DNA profiling has changed a lot, and in the first 20 years changed a considerable amount. And I just want to highlight three different things that have contributed to where we are today. First of all, the introduction of the polymerase chain reaction, or PCR, in the early 1990s meant that the sensitivity of DNA profiling tests was improved. And instead of using a stain the size of an old 50-cent piece, we now profile samples that we cannot see. The second innovation, the second change, was the introduction of short tandem repeats. These are small regions of DNA that can be measured and that vary between people. And it was this implementation of this that enabled the formation of DNA databases. And New Zealand was the second national DNA database in the world. The third example is the introduction of YSTR DNA profiling in 2006. This was important in being able to analyze very small amounts of male DNA in a large female background, and is particularly useful when in looking at sexual assaults. So where are we now with DNA? So first of all, we have autosomal DNA profiling, which is highly discriminating and we use for most of our samples. You have a subset of this, mini STRs, which have been designed to be particularly useful for very degraded samples, such as you might find in a bone sample. STRs we've already talked about. We also have elevated cycle number methods, which we use for the very smallest of samples, for example, fingerprints or matchsticks. We also have a laser microdissecting microscope, which enables us to individually cut sperm cells from a sample and then carry out DNA profiling on as little as 20 or 30 cells. And we have methods to investigate kinship and paternity and familial searching. A brief snapshot of the current status of the New Zealand DNA Profile Databank. There are currently over 200,000 profiles from individuals on this databank, and there are nearly 50,000 profiles from unsolved crimes on this database. And when these two are matched together, which happens more or less every day, 71% of those crime samples will link or match to a profile on the National DNA Database. In addition, almost a third of these crime sample profiles will match to another crime sample on the crime sample database. More recently, we've implemented a YSTR crime sample database where we have unsolved crime profiles generated using YSTR tests alone, and we have almost 600 of these currently. So what are we working on right now? First of all, I want to talk briefly about reporting. So we use something called source and activity level reporting, which is an assessment of the strength to be attached to the findings and the context of alleged circumstances. And in this example, we're going to talk about what body fluid did the DNA come from. So we express our results as likelihood ratios, and that is a ratio of two probabilities. Firstly, in this example, what is the probability of the evidence if the stain came from blood? And what is the probability of the evidence if the stain came from something else, a different body fluid? And we evaluate these propositions using what is called Bayesian networks. And on the slide, you will be able to see an example of a Bayesian network for this exact question. The test results, the evidence, are presented in this matrix, if you like. All the different chemical tests and biological tests that have been done, the strength of the findings, the appearance of the sample. And this probability has been calculated using false positive and negative rates for each of these tests, generating a likelihood ratio for the propositions that you can see on the slide. This is fairly straightforward for blood, semen, and saliva, but now we need to know how did the DNA get there and when did the DNA get there? And this requires more complex modelling of transfer, persistence, prevalence, and recovery of DNA and body fluids that are relevant to the case at hand. And that is what we are working on now. No forensic biology slide is complete without a discussion on genome sequencing. And I think we're all very aware of what genome sequencing is right now. So we have 4 active areas of research in the laboratory at ESR and the University of Auckland. First of all, we have Bethany, who is working on whole genome mitochondrial DNA sequencing for forensic applications. Mitochondria are very useful when the DNA or the sample is compromised and the amount of DNA is low. We have Ryan, who has been sequencing many of the STRs we currently use, plus extra ones, and in addition, DNA markers or SNPs for ancestry and phenotype, in this case, hair and eye colour. He's also evaluating a larger panel of SNPs for kinship testing. Rebecca has been working on chemical modifications of DNA that allow us to distinguish between twins and also to establish the biological age of samples. And she has done this for blood, for semen, and for saliva. And lastly, we have Olivia, who is working on nanopore sequencing. So capitalizing on our work in the COVID sequencing response, we are now using nanopore sequencing to develop methods for rapid on-site testing, sample preparation, bioinformatics, data analysis, environmental DNA, and CITES protected species. Nobody does anything on their own. And this is my team at ESR, to whom I'm profoundly grateful for all the support that they've given me over the years. I'd like to thank again ESR and the University of Auckland for providing me with the infrastructure in which to carry out my work. Ngā mihi o te whakarongo mai, kia ora.